Wonderful. Thank you for the introduction, Robert, and thank you, Christina, for such a great talk. We're super supportive of my Green Lab, and it's great to see that we're getting out more. So without further ado, I'd love to jump in here and talk about caching. So as Robert said, this is our proprietary chemistry that stores DNA and RNA at room temperature for long periods of time. Before we jump in, I just want to calibrate this talk a little bit. So I think Robert is going to open up a poll that we'd love to just get a quick pulse of what the audience makeup is. So the first question is, how many freezers do you currently have? And the second question is, what is your role within your biorepository? And of course, if you don't work at a repository, definitely let us know. So I'll just let that sit there for about 10 more seconds, let people answer. Oh, interesting. So I'm not sure if you all can see the results, but it looks like um the mode here is 2 to 5 and greater than 10 freezers. So it's great to see that we have such a spread. And it looks like only a few of you don't work at a prior repository, but we are going to talk about some economics here. So hopefully that will be of interest. So thank you all for answering that. That's super helpful for us. And with that, I'd like to jump into the talk now. I'm planning on this being a somewhat short talk, leaving plenty of room for questions at the end. So this talk is broken down into 4 main sections. So first, why you should consider ambient storage. We've already heard from Christina a lot of really great sustainability reasons. So we're going to dive into some of the other reasons you may want to consider moving away from freezers. And then we'll dive deep into what is caching, how does it work, when should you consider using it, and then how does caching perform? Does it modify your samples? Does it actually preserve your samples for long periods of time? And then finally, as I mentioned before, we'll take a deep dive into some of the economics and benefits that your organization could see if you were to switch over to caching. And so with that, well, let's jump right in. So when you think about storing your samples, and it seems like almost all of you have a good number of freezers in your institution, there's a number of things that you have to do. And again, Christina touched on some of these maintenance considerations where you're defrosting the freezer, you're cleaning out the filters. This takes quite a lot of time and coordination, especially with the number of freezers you have. Some folks do some pretty complicated things. So this can be quite a challenge. Monitoring and responses is also an important consideration. We've talked to folks who have had to come in on weekends and after hours and had rotating on-call shifts, which can get quite logistically complicated, again, as the number of freezers increases, but the staff normally stays the same for a while. The other thing that we wanted to mention that I don't think has been covered before is this idea that most folks actually make redundant aliquots to prevent freeze-thaw cycles. So when you have a sample of RNA, DNA, or really any sample for that matter, that is frozen, when you thaw it, that thawing process degrades the nucleic acid sample that's kept in the freezer. So if you're constantly taking it out, taking an aliquot and putting it back in, you're degrading that sample over time just by the nature of taking it in and out of the freezer. And so one of the things that people do to combat this is they split their sample into many different aliquots. Say for the math's sake, they split it into 5 aliquots. They put those 5 in the freezer. That way, whenever you need to access the sample, you're only taking one portion, the 20% of the sample in and out and degrading it at a time. This can be a pretty effective method. However, as you can imagine, what you're actually doing is ballooning the number of samples that you need to keep track of and that you need to do this maintenance on and buy freezers for. And so these aliquots can create quite the logistical hurdle, even if they're solving a scientific problem. And then finally, there's emergencies and accidents. I think most famously, we all remember approximately 1 year ago now when a janitor heard annoying alarms and turned off a freezer, destroying 20 years of school research worth $1 million. And things like this happen all the time. This is just a collection of screenshots, some from the media, some from ISPER forums or LinkedIn posts, or in an institution I used to work at where somebody had several -80s go down due to heat and humidity and is just begging to see if anybody else's -80s work so that they can put a few of their samples in somebody else's freezer. So these things happen all the time, and then it can create quite the burden. But aside from that, even if you have a freezer that is working perfectly, That brings us to the second main category, which is running out of space or electrical capacity. So we've talked to folks who have no more physical space in their facility for any more freezers, and they're forced to discard samples or not take in any more samples. And we've also run into folks who had this problem, got a new facility built, but then as they were filling that facility with freezers, they ran out of electrical capacity and they were no longer able to install any freezers. Now, the last thing I want to talk about is considering ambient storage as just a frankly better way to store your samples, or is at least concordant. So one of the main causes of degradation is water and air. So when you put a sample in the freezer, you're not really removing water or air, and really it's the oxygen within those that's the problem, that's degrading the nucleic acids. What you're doing when you're freezing it is slowing down those reactions with the temperature, but you're not really getting rid of the cause of the problem. And with some of these ambient storage solutions that we're going to talk about, you can actually eliminate the oxygen from the environment of the sample, which is a pretty interesting feat, and we'll talk more about that in just a few minutes. Now, some other folks are currently storing their samples at room temperature by essentially storing the primary specimen in FFPE. So this is fixed formalin paraffin embedded. So this is a chunk of tissue that's kept at room temperature, fixed in formalin. And when you're ready to analyze the nucleic acids, primarily DNA, because RNA degrades very quickly in FFPE, you then remove a portion of that sample, you do DNA extraction. But the unfortunate thing is that DNA, while it's been sitting in that FFPE, is degrading about 2 times faster than if you had just extracted it and then kept it in the first place. So before we go on and we talk a little bit about what alternatives there are to this, I'd love to take just one last quick poll. I promise this is the last one of the session, and I'd love to know how you currently store your nucleic acids. And so I think Robert's going to open up another poll here in just a moment. There we go. And again, I'll just leave this up for about 10 seconds. Wonderful. So it looks like the vast majority of you, 79%, are storing your nucleic acids in -80 freezers, which is pretty typical. And then the next largest categories are liquid nitrogen freezer systems, or you only keep the primary specimens, such as FFP or blood. This is pretty typical. But this is really interesting and definitely makes me glad that Christina was talking about freezers and the importance of picking a good freezer. And I'm very excited to continue to talk about maybe some ways we can move away from freezers. So again, thank you for filling out that poll. That's very useful for us to know. So if you were to consider moving away from freezers, there are some commercially available products available. And they are broken into largely 4 categories. So those categories are additives, lyophilization, which is just another word for freeze drying, microscale encapsulation, which is what caching is, and then macroscale encapsulation. So just quickly running through this. So additives are essentially usually sugar mixtures. You pipette that into your extracted nucleic acid sample, and then you dehydrate it. So this process essentially shrink wraps the nucleic acids to the bottom or sides of the tube and protects it from degradants. When you want to recover your sample, you then pipette in some liquid, typically something like water, and you rehydrate it. So you let it sit for a little bit, and then you go on your way. The downside of this is that you cannot take aliquots. So what I mean by this is, can you take an aliquot of that sample without disturbing the rest of that sample? And so here, no, you have to rehydrate the entire sample, take your aliquot, and then dehydrate it again. So you're not able to take your aliquot and leave the rest undisturbed. So that's what I mean by no, you can't retrieve aliquots. So this can be a very cumbersome process, because every time you want to retrieve a part of the sample, you're having to go through this entire process again. And we've actually heard from some people that even though they were excited to use this method, they ended up not implementing it because of this pain point. And then when you look at the data and you ask, does it actually slow degradation? The answer is only in very low humidity conditions that are well sealed. We've heard of folks who saw some exciting preliminary evidence in low humidity conditions and then implemented it into their lab, which was not as low humidity. And the sample was basically completely degraded within a couple of years. So this was not ideal and excludes many people from considering this solution. And then this is not a particularly scalable solution. The throughput is rather low because of that whole process of dehydrating, rehydrating, and then not being able to take aliquots. So that brings us to lyophilization. I'm sure most of us have worked with samples that have been lyophilized before, whether they be primers or something else that you're receiving, or maybe your institution lyophilizes some samples. And so if that's you, you know that the complexity to this method is quite high. It requires some complex configurations, different configurations for different sample types, and it also requires specialized equipment because of that freeze-drying process. But again, the recovery method is quite simple. It's a simple rehydration. Again, you can't retrieve an aliquot, leaving the rest undisturbed. You have to rehydrate the entire sample and then go through the entire lyophilization process again with the remainder of the sample once you're done taking your aliquot. This method, however, does slow degradation because it does not involve any sort of sugar matrix. It's just the sample that's been freeze-dried. It does seem to offer more protection. However, you do still need a very well-sealed sample to protect it from any humidity that's in the environment. And the half-life is likely something on the order of decades. However, research into this is still ongoing. And it's potentially scalable. So traditionally, this has very much not been scalable. This specialized equipment takes up quite a bit of space and is not high throughput, but it's looking like some emerging technologies are starting to change this, but even still, the throughput is relatively low. And that brings us to encapsulation, which here we split out into micro and macro. So we'll go into much more detail on this in just a slide or two. But microscale encapsulation is what Cache does. And essentially, this is on a micron scale, encapsulating your nucleic acids in a protective shell. And so this is medium complexity. It's a multi-step process with just simple pipettes. And again, with these simple pipettes and standard lab equipment, this means that you aren't buying any specialized equipment, and you can likely already do this process with the equipment you already have. The recovery method, instead of rehydration, is de-encapsulation, so breaking open that protective shell. And this takes only a few minutes. It's actually comparable with taking a sample out of a freezer, and it can be even quicker than that because with a freezer, you're having to slowly thaw it for 30 minutes oftentimes. And as Christina said, sometimes folks really struggle to find their sample within a freezer, especially given ice buildup. That can be a massive problem there. That is not encountered with encapsulated room temperature storage. So the important thing is this is the only method that can retrieve aliquots. You can retrieve just a fraction of the encapsulated samples, leaving the rest undisturbed. Because this is on the microscale, you have many, many thousands of encapsulated nucleic acids in a reaction. So you can just take an aliquot, leaving the rest undisturbed, and you don't have to go through this encapsulation, de-encapsulation process multiple times per sample. That's a one-time thing. So that's very exciting to folks. The other thing is that it does slow degradation because it has that protective shell around it. It is able to protect the sample from the degradants in the environment on the order of decades. And again, I'll go into that data more in just a few slides. And then finally, this is a scalable operation. I'll talk more about this later, but this is automation compatible. And with just a very simple liquid handler with a 12-headed multi-channel pipette, we're able to store over 100,000 samples per year. And if you have something larger, that number goes up to well over 500,000. So that brings us to macroscale encapsulation. So macroscale encapsulation has a few downsides. It's high complexity. And if you want to encapsulate these samples with this macroscale encapsulation, which is a metal capsule about 20 millimeters high, you either have to ship it to the company that offers this service, or you have to put considerable time and resources into setting up a very specialized one-purpose infrastructure. But again, de-encapsulation, it's breaking open that metal capsule and rehydrating the sample within. Fairly straightforward. Again, because it's at this macro scale, you can't retrieve an aliquot leaving the rest undisturbed. You need to go back and forth between de-encapsulation and encapsulation every time you want to access part of the sample. But the upside to this is that it does slow degradation considerably with a half-life of thousands of years. So if all you care about is storing your sample to the end of time, this is a great option, but unfortunately it's not scalable, it's very expensive, and it's very difficult to implement. So with that, we are very excited about caching this microscale encapsulation because we feel that it is the best option for the greatest number of labs. And so with that, I'd love to dig into what is caching exactly and how does that work? So caching starts with a proprietary bead that we've designed. This bead has a very slight charge to it and it's silica-based, so it's very non-reactive. And when you put those beads into a solution with nucleic acids, the nucleic acids are attracted to the bead and weakly bound to it. Then you pipette in a couple of encapsulants, and these form a matrix that builds up over time to form a protective shell. And this is what allows us to keep these samples at room temperature for long periods of time. So at that point, you're ready to stick this on a shelf somewhere and come back to it months, years later. And when you're ready to come back to it, you can then take an aliquot of the sample, leaving the rest completely undisturbed. Those are still encapsulated. You put them back on the shelf. And with this aliquot that you've taken, or you can take the whole sample if you wish, you then pipette in an etching solution. This etching solution etches away the protective shell on the outside, which is silica-based, and the inner core, which is also silica-based. But it leaves the nucleic acids untouched and undisturbed. And so now your nucleic acids, after just a few minutes, are free floating in solution again, and you can continue on to your downstream assays. So some of these assays, we do recommend that you do a desalting step, something like sequencing library prep, but other applications such as qPCR or PCR or just doing tape station, you do not need to do the desalting step. So I've linked a couple of publications should you wish to read more. But I'd love to take a step back now and say, okay, now that you have a little bit of a good understanding of what caching looks like, how does this fit into a workflow? So essentially, if your organization starts with any sample type, that could be cancer cells, it could be bacteria, viruses, it could be plants, we only care about the extracted nucleic acids. And this could be DNA, RNA, total nucleic acids, where both are floating in solution. We're completely agnostic. Once you get to this extracted nucleic acid stage, you add in the beads, you then add in the encapsulants, then you have your cached sample and you store that at room temperature for many years to come. And so once you are ready to retrieve this, as I mentioned before, you take the aliquot or your whole sample, mix that in with an etching solution, and then you're free to go on your way. So when you think about the whole end-to-end system at your biorepository, caching is a very modular part of that system that can fit in and replace say the freezing that you're currently doing, leaving the up and downstream completely undisturbed. So, as I mentioned before, it was very important to us to make sure that caching was something that you could easily implement in your lab at any scale. And so while this can absolutely be done on an individual scale with one pipette and just a few tubes at a time, you can also use multi-channel pipettes with any liquid handler. So here we've broken it down by the numbers, and we have our director of automation who has created an application note detailing what these protocols are and a demo video that's available as well, showing in real time what this process looks like. In our lab, we commonly use a 12-channel Integra Assist Plus. This is a very simple instrument shown here in that screenshot of the demo video that costs on the order of about $30,000. And with just this system, encapsulation takes 32 minutes total, with only 3 minutes of hands-on time. And that does not include the incubation process, which is 48 hours shaking at room temperature. And you can actually see in this demo video, there's this shaker on the right that actually shakes multiple 96 well plates at the same time. And so again, this is a very hands-off process. It's just, you just let it shake at room temperature for 48 hours. And then the sample is fully encapsulated by the end. Now, when you're ready to de-encapsulate, this small-scale system takes 30 minutes. And again, all these times are for a 96 well plate. The hands-on time, again, is very minimal, only 2 minutes. And the total throughput of this is over 100,000 samples per year. And that is just working 4 days a week, 8 hours a day. This is, no overtime, very manageable numbers that can be pushed up should you need. And then if you need even greater scale, you can use any 96-channel system that drops the encapsulation and de-encapsulation times considerably and enables you to have a throughput of well over 500,000 samples per year. So we're very excited to be able to offer this technology at truly any scale that your organization may need. And now let's go on to the next part of the talk, which is caching performance. So we completely understand how valuable these samples are. The last thing we would ever want to do is to be modifying these samples in any way. You can think of caching like a pause button where your sample comes in, you cache it, and you're basically pressing pause on the degradation until you're ready to retrieve it again. We do not do any modifications to cache it, and the process of caching itself does not incur any modifications. So here we're presenting data on shearing. So on the left, you can see that we're reporting integrity numbers from a tape station. So these are DIN and RIN scores, if you're familiar with that. So on the left, we have genomic DNA, and in pink, you can see the pre-encapsulated scores. And then in green, right next to it, you can see the scores after they have been cached and de-encapsulated and filtered. And so this is essentially the moment that you could do truly anything downstream. And you can see that those scores remain very high. And this is the same holds true for RNA, where when you compare the pre-encapsulated scores to the post de-encapsulated and filtered, you still see very high scores there. And on the right, we show the traces themselves from the bioanalyzer. And so you can see that before caching, the genomic DNA has this peak at well over 60,000 base pairs, and the same peak is seen post-filtering. And with the total RNA, you can see that the peak distribution and intensities are nearly identical between the RNA before it's been cached and then after it's been cached and filtered. So this was incredibly powerful for us to see, and demonstrates that caching is not shearing the nucleic acids at all. It's leaving these samples intact for you to analyze, even with long read sequencing for years to come. So the next thing that we did was we partnered with UHN and the Princess Margaret Cancer Center, and we took extracted nucleic acids from FFPE samples. And what we did here was we sequenced them with next generation sequencing, and we wanted to make sure that caching did not modify the base level nucleic acids themselves. Even though there's little to no shearing, that doesn't do you any good if the variants called are different with pre and post cache samples. So what we did here is we took these FFPE-derived nucleic acids and we split them in two. We froze half of the sample and we cached half of the sample. And so then we looked at concordance between the variants that were called. And so ideally, you want to see a genotype concordance close to 1. And when we look at the difference in concordance between the technical replicates, we see that there is naturally some variation in concordance, which is to be expected with these systems. But we actually see even less variation and higher concordance when we compare frozen samples versus cached samples. And this holds true for both SNPs and indels. So what this is showing us is that there is no nucleic acid modification at the sequence level between cached and non-cached samples. So this is very exciting for us. And it's showing you that truly caching is not having any measurable effect on the sequences themselves. This also had a really nice benefit because we were working with FFPE-derived nucleic acids. FFPE is very notorious for being a messy sample type. It has a lot of contaminants in there. And that was also a question of, can you cache such a messy sample? And the answer was yes, and with no problems. So again, caching is compatible with a wide range of inputs. And this is something that we wanted to test even further. So what we did here is we took DNA samples that were both long, over 60,000 base pairs, and short, only 130 nucleotides total. And we did the same with RNA, long and short. And what we found is that with caching, no matter if it was DNA, RNA, short, or long, we were able to get consistently high retrieval rates. So the amount of sample that you put in compared to the amount of sample that you pulled out was very close to 100% in all cases. So this was something that we were excited to see, but we also wanted to make sure that the inputs weren't so constrained that the caching became difficult to perform. So what we did here is we did an experiment where we cached very small samples that were only a few hundred nanograms, and we also cached samples that were closer to 3.5 micrograms. And using the same protocol, we were able to again get very consistent high recovery rates, close to 100%. So this is very exciting to us. We don't recommend that you cache samples lower than 100 nanograms. This is certainly fine, but then we find that you actually start losing a measurable amount of your nucleic acid to the tips and the tubes. So again, wide variety of inputs. I would generally recommend over 100 nanograms, but for most people, this is totally fine. And we have some partners right now who are testing some lower concentration samples as well. So we're very excited about that. And finally, that brings us to long-term preservation. So one of the most important things about preserving your sample is making sure it will still be there when you want it. And we've found that with DNA, we have a half-life at room temperature at 65% relative humidity with a half-life of decades. So just to give you a little bit of background, I know Robert said I have a PhD in computer science. My PhD was actually using molecules to do computation. And one of the ways that we wanted to do that was by storing DNA for long amounts of time and then doing computation with them afterwards, after decades or hundreds of years. And the only way this made sense was to store these samples at room temperature. And so I went out and did a lot of literature searching, and I could not find anything that compared these different methods head to head. So what I did in this figure, which was published before Cash was even a company, before I had even heard about Cash, was I directly compared a bunch of mostly commercially available products. And so at 50% humidity, you can see that at room temperature, which is where that vertical line is at 20 degrees Celsius, most of these conditions, which are represented in a solid line, have a half-life of far less than 10 years. So this is pretty devastating. Obviously, you do not want to be storing your samples with these methods that only last a short amount of time. So we repeated this experiment with cached DNA. This is shown in the dotted teal line right here. And we find that at even more unfavorable conditions, at 65% relative humidity, we're able to still have a half-life of decades. So this is very exciting, providing very good evidence that caching does protect your samples for long amounts of time, and those samples are degrading quite slowly. So how about RNA? We are in the process of doing a similar experiment for RNA, but what data we do have in the meantime is showing RNA at room temperature in real time. So what we did is we had a pool of RNA and we split it into frozen and cached. So the frozen samples, we froze a bunch of different aliquots. And so every time we went to take a time point, we pulled those aliquots from the freezer and they had undergone no freeze-thaw cycles. We wanted to be as fair as possible in this comparison. At the same time, when we took our cached samples, we dehydrated them, then rehydrated them, took an aliquot, and then dehydrated again. So we didn't go in and out of the caching process, but we did dehydrate, rehydrate. We wanted to see if perhaps our claim that you can take an aliquot and leave the rest of the sample undisturbed was true. And that is indeed what we found. We found that over the course of 2 months, the cached DNA had no measurable degradation. And so this was very exciting to us. Again, this is RNA kept at room temperature for 2 months with no degradation. And so for a lot of people, this is a pretty big game changer for them, allowing them to more seriously consider storing RNA. So I hope by now I've convinced you to at least consider caching from a scientific viewpoint. But now let's switch gears a little bit and talk about what this might mean for your organization. So when you think about implementing cryostorage at your organization, typically there's massive costs due to energy consumption. You also have these HVAC costs. Christina talked a little bit about that as well in her presentation right before this. They also take up considerable amount of space, require a lot of maintenance, and of course the cost of the freezers themselves is significant. But if you were to switch over to caching, these or any ambient preservation technology, this changes considerably. So with caching, this green section is completely eliminated, and these gray sections are reduced costs. And so with caching, you have no ongoing electricity costs. It requires considerably less space, again, because you're not exploding the number of samples you're keeping track of with the aliquots, but you're also not requiring these bulky freezers anymore. There's considerably less maintenance because you're no longer maintaining any of this equipment. The CO2 emissions are drastically reduced. And if you ship your samples, right now the standard practice is to ship your samples on dry ice with overnight shipping to make sure that you still have dry ice when it gets to its final destination. But with caching, you no longer need to do any of that. So the cost goes down considerably. Now, related to this is this idea of compliance and insurance. Because you no longer depend on the electrical grid, you can free up a lot of your time and energy, no longer worrying about a typhoon that's coming in, or the power going out. So with no more considerations for backup power or temperature monitors, this frees up you and your organization to focus on other things. And as I've already mentioned before, retrieval with cached samples is quite quick, and it can actually be faster than freeze thawing. And with the ability to do selective aliquoting, leaving the rest of the sample undisturbed, you're able to get more out of your samples. And as I mentioned before, this method is also compatible with both manual and automated workflows. So again, allowing you to cache at any scale your organization may need. So as we've shown here, we have an impact calculator that's linked in these slides that you can access on our website. So it allows you to put in more details about your organization and see what benefits caching could bring to you directly. And last, I'd love to talk briefly about a case study. So we interviewed a company. Here we've anonymized them by calling them Nucleic Therapeutics. And essentially their biostorage operation costs look, quite frankly, like a lot of you in the call today, based on that survey that we gave you at the beginning, where most of you, or a lot of you, had over 10 freezers. That's almost exactly what this company looked like. They had many freezers. Their annual biostorage operation costs were $78,000 per year. So over a 12-year cumulative lifetime, that was almost $1 million. Now, if they were to switch to caching, their biostorage operation costs would shrink drastically, down to $21,000 per year, and over 12 years, down to $248,000. So this is huge. This is 74% cost savings, not to mention the millions of kilowatt hours saved. And as I mentioned before, this kind of disaster proofing, this idea that you no longer need backup power, you're no longer requiring dependence on the grid, and you're completely eliminating dry ice shipping. So this is exceptionally compelling, and I'm happy to take more questions about this after the fact as well. So in summary, caching at your organization can help make your operations more sustainable. And as Christina mentioned in her talk before, sustainable can mean a lot of different things. It can mean energy sustainable, operationally sustainable, or logistically sustainable. It enables your organization to preserve these samples at scale, maybe enabling you to store more or preserve your samples for longer. And it also allows you to do this within a standardized workflow. So the protocols are exactly the same for DNA as they are for RNA, and they're compatible with truly any standard liquid handling system that exists. So while we use the Integra, this can absolutely be implemented on equipment that you already have. And most importantly, we want to bring ambient nucleic acid storage to you. We want to make sure that you're able to perform this at your facility. We can also perform it at ours and store your sample should you need to, but we want to empower you to move away from freezers at your facility. So, in summary, caching preserves both DNA and RNA at room temperature for long periods of time and does not compromise on the quality. Caching allows your organization to store nucleic acids at any scale that you should wish. And we encourage you to join us in implementing a more sustainable nucleic acid preservation method for years to come. And so actually right now, I'm very happy to announce that we have demo kits available for free. So if you go to this QR code or you visit us on our website, or you can email me, we're happy to set up a talk and see if caching makes sense for your organization. And then we can send you a free demo kit. So this demo kit has enough reagents in it to encapsulate 25 cached samples. And then you're able to evaluate in your hands very rapidly whether or not this is a solution that makes sense for you. So I'd encourage anybody interested, feel free to apply for a demo kit, and I'll get back to you shortly. And with that, I'd love to open the floor for any questions that you folks may have. All right, I see we already have. Oh, yeah, Mike, I see that you've agreed to answer this question live from Asim.
Yeah, sorry, Leah. Can you guys hear me all right?
Yep.
Shelby. Robert, sorry, I'm just getting used to this Q&A platform, but looks like Asim's coming on to ask the question. Lee, I think you're the best person to answer that one.
Cool. So Asim, I see that you ask if the temperature is slightly increased to over 20 degrees Celsius and humidity is around 50, how would that impact the half-life of cached DNA? So Right. The data that I showed was degradation at 65% relative humidity. So we would expect that if anything, lowering the humidity would increase the half-life. Now, increasing the temperature is then going to speed up the rate of degradation very slightly, though, from 20 to 23 degrees Celsius. So what we can do, and I'm happy to follow up with you later. We're currently doing some studies that do work at 50% humidity, so we'll be able to get you those numbers. And once we have those numbers, we can then extrapolate to 22, 23 degrees Celsius. So essentially, stay tuned for exact numbers in the next few months. But in practice, I would not expect this half-life to change very much by tweaking the temperature up slightly and then humidity down slightly. I would expect those numbers to stay almost exactly the same with half-life of decades. And I see another question, is there a maximum amount of DNA that can be cached per well? So we have tried up to about 5 micrograms and found high recovery rates. Beyond 10 micrograms, we start to see that we're probably maxing out the bead capacity and we're no longer caching quite as much material. So I would say that your maximum is somewhere around 5 micrograms, but this is still being explored. And that is something you could absolutely explore with the demo kits if that's something you're interested in. All right, and then I see, can cache be applied for preservation of proteins? Unfortunately not. So the thought here is that these beads are quite small. They're on the micron scale, and most of the proteins that people work with are going to completely dwarf this. However, we do have quite a lot of people that are really interested in amino acids. So it's possible that very small proteins or just a collection of amino acids themselves may be able to be cached, but this is not something we've tried. I cannot make any any guarantees about this. Again, if this is something you're interested in, we'd be happy to talk more. But great question.
Any more questions? Oh yes, here we have one.
And Carly, I might pop it over to Mike for this question.
Hey, Carly, great question. Yeah, in terms of the pricing and cost of sample for caching, we like to work with organizations to think about the best implementation for you. So definitely want to get a better sense of any existing liquid handlers you might have and the current setup in your lab. But currently we found that because of the long-term protection that is conferred by our method, we usually start the conversation around $10 per sample. Of course, if you're doing this at a high level, we're more than happy to offer bulk discounts. Our goal is to make this not only a more sustainable solution for your lab, but also a cost-saving measure. And with all the benefits that Lee described, of not having to pay for dry ice shipping and not having to store redundant aliquots due to freeze thaw considerations, we're really confident that this can make a positive ROI for your lab. In terms of the kits, this is something that we aren't charging for right now. We're more interested in getting the feedback from the users. So those are free. You can just request them in the QR code. And as long as you're willing to have a conversation or two with us over Zoom, we are more than happy to ship it anywhere in the world right now.
Okay, any more questions?
I was just going to say, I know Lee mentioned a few resources during the talk. I'm just seeing that there's a link resource section on Zoom. So I'm going to start adding a few of those right now in case anyone wants to check them out.
Okay. Yeah. Could I just say one thing, Lee, that I was really impressed to see that caching is the only one of the 4 different types of ambient temperature storage, which allows convenient aliquoting, which is really impressive. And I guess that's a big advantage over storage in freezers as well, because it's very difficult to aliquot without damaging the sample.
Yeah.
For a
it's certainly one of those benefits that's it's a little bit hard to describe, but once you start to see it implemented, you realize really what a what a big difference it makes compared to everything else that's out there.
Amazing.
Sorry if there was a question in there. I thank you for the kind words.
Okay, so I mean, if you're interested in DNA and RNA, this is definitely the way to go.
I mean, again, I am biased. I work at Cache, but I worked in this field for a considerable number of years before caching even existed, and there is a reason I'm here at Cache. I'm very excited about this method.
Okay, any more questions?
And then I see a question from Mirko of will the presented slides be available for download? And again, this is a question for Mike.
Yeah, I think Robert actually records all of these talks on his platform, so you can rewatch the video, and we're happy to link the slides to our website as well. So yeah. See that
Okay.
Christina and I have populated this resource page with some links. So encourage all of you who are still here on the call to check those resources out regarding the freezer challenge, improving your laboratory sustainability, and requesting a demo kit, which is, I'd say, the clearest action you can take after this call if you're interested in trying the caching chemistry yourself.
Okay, could you just explain, Mike, whereabouts is this resource page that you're completing? Where can we find it?
So I'm curious if anyone on the webinar viewer can see it ourselves. I know for me, at the bottom row, it said resources, and then on the right side, it says links, documents, and speakers.
I got it, okay. Excellent.
Yeah,
Okay.
hopefully people can access that. There's links in there, and then under documents, specifically for Cameron, who asked that question in the last segment about the ultra low temperature freezer study that was done by the SEAL report. I uploaded that there as well. So that's under documents instead of links because it's a PDF. But you can access that there. It's a very dense report.
Okay, great. So just in case anybody hasn't found it yet, at the bottom of your screen, far right, there's an icon that says resources, and that's the place to look. And there are 3 sections. One of them is links, and the other is documents that you ought to look at. Okay. So any more comments? Any more questions? If not, then I guess we should bring this to a close. It's been a fascinating session, and really appreciate the presentations from all of our speakers. So thank you very much indeed.
Well, thank you all.
Thank you.
Thanks, Robert.